Scientists have developed a way to edit DNA using a technology called CRISPR. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats and works as genetic scissors. CRISPR makes it possible to snip out undesired bits of DNA from our cells. This process holds the potential to cure genetic conditions such as cystic fibrosis. However, it seems that doing this isn't without risk. Researchers at the Karolinska Institute in Sweden have discovered that using CRISPR may increase the risk of cancer in certain cells. I went to the University of Cambridge and spoke with biochemist Jussi Taipale to learn more.
Medically, the point would be, of course, to fix things. If somebody has, for example, a gene that is mutated and causes disease, the idea is to go in and change the DNA sequence back to what we call wild type, so the normal sequence, and not this mutated sequence that causes the disease, in order to cure the disease. And mutations happen all the time in our body, and most of our cells have large numbers of mutations. And some of them are more likely to turn into tumors than others. But if we now take a large number of cells and we try to edit all of them, then there's a risk that the normal cells that are less likely to form cancer die, and we are left with the cells that are already on the path towards cancer. This is predicted to have a potential for increasing the risk for cancer in the patient if this was done.
CRISPR has shown a lot of promise in mutated genes, but Jussi and his team explored the use of CRISPR in completely healthy genes. What they found was alarming.
When we edited the healthy cells, sort of the alarm went off that there's DNA damage, that the DNA is cut, and a protein called p53 got strongly activated and stopped the cell from dividing.
p53 is a protein associated with tumor suppression. Cells that are deficient in p53 have a greater risk of growing tumors and possibly cancer.
Some cells lack the p53 gene, even normally in the human body. But if we now take a large number of cells and we try to edit all of them, and then this p53 gene gets activated, then there's a risk that the cells that already have lost the p53 have a growth advantage, that they grow faster, or that if lots of cells die, it could be that the normal cells that are less likely to form cancer die, and we are left with the cells that are already sort of on the path towards cancer.
The good news is, through further research, we might be able to minimize the risk of cancer.
We tested one cell type, but we cannot possibly as a single laboratory test all kinds of cells, and in particular set up complicated protocols for example, clinical treatment. So our idea was to post this out so that people would be able to see that this could potentially be a problem, and they could test this situation in their context and try to design the experiments and the treatment protocols so that the risk from this source would be minimized.
Although Jussi and his team have identified the risks of CRISPR, they still find it very promising.
It has enormous potential because it's basically a scissor that can cut the genome wherever you want. So it's basically highly effective at cutting the genome. This is very useful if you need to inactivate a gene. Sometimes there is a mutation that causes a defective gene that causes disease directly. And we usually have 2 copies of all genes. So just deleting the defective one often could, in theory, cure the disease. And then you can just cut with CRISPR and destroy the defective copy. So this is a very promising direction. And also this editing where you precisely fix the defective gene is also quite promising. So I think there will be many treatments that are based on CRISPR or some related technologies. It has enormous potential.
Jussi Taipale there, and that study can be found in the journal Nature Medicine.